Hello everyone and welcome. I'm Louise, Curator of Natural History for the High Desert Museum. Thank you so much for attending this Natural History Pub, and thanks to our wonderful museum members for helping to make this whole series possible. We would also like to say a big thank you to our programme sponsors, Fairfield Inn and Suites, and the Pacific Northwest Forest Service Association, also known as Old Smokies, and also the National Endowment for the Humanities. We have tech support available tonight should you have any issues. Thank you as always to our talented communications manager, Becca, for being here to assist. At the bottom of your Zoom window, you should see a chat function if you wave your mouse or your pointer over the bottom area of your Zoom. Use that to ask any questions you have throughout tonight's talk, and then at the end of the presentation, we'll select your questions from there. Without further ado, I'm absolutely delighted to introduce our speaker tonight. Dr. Katrina Moser is a professor in the Department of Geography at Western University in Ontario in Canada. She gained her Ph.D. from McMaster University in Ontario and then worked for the University of Utah for 8 years, hence her work in the Uinta Mountains. Katrina's research is focused on water quality and quantity issues, and specifically how humans are impacting these important resources. Key questions that she is interested in are: how will future climate warming impact water resources? How has anthropogenic pollution affected pristine alpine and Arctic lakes? And can climate and pollution synergistically impact water resources? In order to look at these questions, Katrina uses paleolimnological techniques, which provides her with long-term records, hundreds to thousands of years of changes in water quality and quantity. Paleolimnology uses fossils and geochemical signals preserved in lake and river sediments, also known as mud, to infer environmental change. Welcome, Katrina, and thank you so much again for joining us tonight.
Well, thank you very much, Louise. That was a very nice introduction. Thank you very much for hosting me. And thank you to the audience for attending tonight. All right, can you see that all right, everybody? Yes, okay. So the title of my talk is Still Waters Run Deep, Mountain Lakes in a Changing World. And the purpose of my talk is to show you how records archived in mountain lakes, or mountain lake sediments rather, can be used to address questions about global change. And I'm going to take you on a bit of a journey tonight, and we're going to talk first a little bit about mountain lakes and global change. And then I will tell you a bit about paleolimnology and how we can use paleolimnology to study global change. And I'm going to use the Uinta Mountains, where I've been working for about 20 years now, to show you some examples of how we actually get at global change through lake records. So first, mountain lakes and global change. So when I say global change, I'm referring to environmental change that's happening on a global scale. So a really good example of global change is climate change. Humans have increased the amount of CO2 in the atmosphere, and that's causing global temperatures to rise. But there's lots of other examples of global change. For example, we might have a mass extinction event. That would be a global change. Or we might think about how agriculture and using synthetic fertilizers has impacted terrestrial or aquatic ecosystems. Those are all examples of global change. And mountain lakes are particularly useful for looking at global change for a number of key reasons. First of all, you find mountain lakes globally, so they cross latitudinal gradients, which allows us to get a global perspective on a particular environmental issue. Mountain lakes are really sensitive to environmental change, and steep elevational gradients result in distinct environments that lie in close proximity to each other. So within a single watershed, that allows us to look at how different environments might respond to the same change. So this is just a map of the world, and shown in gray are mountain ranges. And you can see they extend from the very most southern latitudes to northern latitudes. And the blue circles are showing you where there's been lake studies done within mountain ranges. And the bigger the circle, the more lakes that have been studied. So there's definitely concentration in sort of temperate or less tropical latitudes. But nevertheless, there's lots of scope for us to look at global change, lots of places that we could be looking at lakes to get, or mountain lakes rather, to look at global change. This is an image from the Uinta Mountains, and I use it to show you what I mean by steep environmental gradients. So in this image, you can see closed forest, and you can track it going upslope in the distance and see it transition into true alpine tundra. And you know, if you've hiked in the mountains, that if you start out at low elevation and you start hiking up, you cross temperature or you feel temperature variations. And this is as a result of steep temperature gradients that you're crossing. So this means that in alpine areas, if we increase temperature a little bit, we will see shifts in those temperature gradients, and in response, we'll see changes in vegetation. So this is what I mean by the system is sensitive. These steep environmental gradients mean the system will respond relatively quickly to any change. As well, within these environments, you can see again in this picture, it would be possible to look at a lake in a closed forest and compare it to a lake in an alpine tundra setting within a single watershed to see how lakes in different starting points in different settings respond to the same stressor. So again, this is a real advantage to using mountain lakes to studying environmental change. The other thing I want you to note in this image is that there's relatively little direct human impacts occurring within this setting. Mountain lakes, we tend to think of them as pristine. I'm going to suggest by the end of the talk, this probably isn't quite correct, but certainly there's limited anthropogenic activity occurring in the catchments themselves. What that means is that these lakes are more sensitive to global stressors, things that aren't happening on just a local scale. So again, this picks up on this idea of sensitivity. So how can we study global change from mountain lakes? If we want to look at global change, we really need to have a long series of measurements. And if we're interested in lake properties, it's very rare to have a record of measurements that extends more than a few years to maybe a few decades at very most. But if we're interested in global change, we really need to have long-term records that might span decades, hundreds of years, thousands of years. So how do we get those? And this is where paleolimnology comes into play, or using lake mud, basically to track changes. And paleolimnology offers us two key things. It offers a historical perspective, this long-term record, but it also gives us a holistic picture of environmental change. And this is just to show you, this is what we're going to be talking a lot about tonight, is lake sediments collected in this sediment core tube. And anyway, I'll come back to that. So lake sediments, they are basically archives of the past. They're laid down year after year, layer after layer. And in this image, you can see two scientists trying to collect a core of mud. All right, so using a system that I'll show you in a minute, they're retrieving the sediment here. And these sediments that are accumulating over time, they accumulate continuously. And they continue such that the oldest is on the bottom and the youngest is on the top. And we can date using radiometric dating, which I won't talk too much about, but that gives us the absolute ages of sediments. So how do we get those sediments out? We have a variety of coring devices. Some, like this one in this image, are driven into the sediments by gravity. So this is just on a rope. It would be lowered down and gravity would allow the tube to penetrate into the sediment. Sometimes they are driven in using core rods, and that's what you're seeing in this image is 3 of us trying to drive a core tube into the sediment. They all work on the same basic principle though, that what we do is once the core tube's in the sediment, there's a plunger that you can see here that is triggered from the surface. The core tube is in the sediment. We trigger this rubber stopper that slides into the top of the tube. And just like if you were stirring your drink with a straw, put your thumb over the top and lifted it up, you create a vacuum and keep the material in the tube. And this is what you see here is the mud after it's been extracted from the tube. And you can see, if you look sharp, there's a sort of pink clay here and then more organic sediments on top. It's beautifully preserved. And this would be the oldest down here, and this would be the youngest up here. So we can collect these sediments, and as I said, date them, and that then provides us a record of change over time. So this is our historical perspective that we have in these lake sediments. And anybody can see, you don't have to have a PhD in paleolimnology to see that there's a change in the sediments here. So this is a smaller tube, but here's the top, that would be today, that would represent today. This is a few hundred years ago, and you can see here something happened to cause a change in the type of sediments being deposited. And within those sediments, there are fossils and biogeochemical signals that we can use to tell us about how the environment's changed over time. These are both cores taken from the Uinta Mountains. This one's actually from Great Salt Lake. But again, you can see these changes in what's being deposited. That tells us a lot about what the environment was like in the past. So why do we want a historical perspective to study global change? It provides us with baseline conditions. It tells us what the conditions were like before disturbance and after disturbance. It also tells us about natural variability. How much does the system normally vary? It tells us about trends and long-term trends. So are we moving outside of natural variability, or are we still within natural variations? And it allows us to compare to historical events. So if we have a dated core and we see that there's a long-term trend, when did that system start to change? And is it related to some known historical event? So that's the beauty of these lake sediments. The other thing is that they provide us a holistic picture of global change. So you might think, oh, we're getting mud from the bottom of the lake. That's going to tell us about the lake itself. And you'd be right. It does tell us about the aquatic system, but it also tells us about the terrestrial system and the atmospheric system. So we get things falling into the lake from all 3 systems, and they're recorded over time. So if we know where something's come from, we can tell about not only how the lakes changed, but how the atmosphere's changed or how the terrestrial systems changed. How do we get these changes? What do we look at? So what you see here, this is another Uinta Mountains sediment core. And over here, you can see I'm sectioning it slice by slice, half centimeter by half centimeter. And within each of those slices of mud, there's all kinds of fossils, biogeochemical, geochemical signals that we can use a bit like a detective uses fingerprints to figure out what the environment was like in the past. Now, there's a lot of different proxies we use. The proxy that I use the most is something called diatoms. And that's what you see on the far right. Over here are pictures of diatoms. And diatoms are microscopic. So for example, this is probably about 10 microns. And of course, 1 micron's a 1/1000th of a millimeter. So very small, like smaller than the diameter of your hair. And These diatoms are a type of algae. They're single-celled, and they are characterized by a cell wall made out of silica. So you could think of it as glass. They're like little glass pillboxes or petri dishes. And when these algae die, these cell walls are preserved in the sediments, and they're taxonomically unique. So what I mean by that is each species has a very different looking cell wall, and you can see some of the variations here. These are 3 different species. They occur in huge numbers, so millions and millions of them in any lake. They're found in pretty much any place you have water and light. You will probably have diatoms. And because they're made out of opaline silica or something very similar to glass, they're very well preserved in lake sediments. So the last thing that's important about diatoms is each species has very specific environmental conditions that it needs to live. So some like water's more acidic, some like it more saline, some like it colder or warmer. And so depending on which species we find, it tells us a lot about what the environment was like in the past. So this is one of the proxies that I use quite a bit, and I'll be talking about today. But there's a whole toolbox of paleo indicators or proxies, things that tell us about the past. And these are a few pictures you can see. This is a diatom here. These are chrysophyte scales, which is another type of algae. Little Cladocera, and you find their carapaces in the sediment. This is a chironomid, which is the larval stage of a midge, so the non-biting midges. They look just like a mosquito once they change from their larval stage, but they don't bite. But in their larval stage, they have a chitinous head capsule, which is well preserved in the sediment, and that's what you see here. And you can identify them based on their teeth structure. We also have terrestrial sediments, geochemistry, which can be related to atmospheric deposition of dust, or it can be related to the terrestrial system, or it can even be related to precipitates in the water itself. The other thing that we use a lot are isotopes, and I will talk a bit about isotopes today, stable isotopes. Different than the radioactive isotopes we use for dating, stable isotopes can tell us about climate, about nutrient sources, and a whole suite of other things. And I'll talk a little bit more about isotopes down the road. So that's paleolimnology. We talked a bit about mountain lakes and global change. And now I'm going to tell you a story, take you through my time in the Uinta Mountains and look at some global change problems and what we've learned from the Uinta Mountains. So this is where the Uinta Mountains are in Utah, right on the border of Utah, Wyoming, and Colorado, just to the east of Salt Lake City, just to orient you. And this is the mountain range itself. I'm going to be looking at actually 9 different lakes today. The yellow stars show 6, and then there's 3 others that are over here on the north slope. So these are all on the south side of the Uintas, and then 3 over here on the north side. So the story starts, as I said, about 20 years ago, and I had just taken my first academic job at the University of Utah, and I was busy collecting samples from lakes in the Uinta Mountains. And I went back to my lab and I started analyzing them, and I was surprised to see these diatoms show up in the surface sediments from lakes that looked like this. And why was I surprised? So this is a diatom called Asterianella formosa. And we find it in temperate environments, usually in really polluted lakes, like lakes that have very high amounts of nutrients in them. So I was like, well, why is this here? This lake does not look like it should have very much nutrients in it. So that puzzled me. And that basically started me on my journey in the Uinta Mountains. So the first thing we did was to take a look in a few lakes and see when did this diatom show up, or has it always been here? And this is what you're seeing. This is just a picture of the Asterianella formosa. Here's present day, and this is going back to 1450. And as I said, these dates are based on lead-210 dates and also radiocarbon dates. And what you'll see, this is just basically, you can think of this as the amount of Asterianella formosa. Each of these little bars is telling you how much Asterianella formosa is in the sediments at different times. So before about 1650, there was basically none. And then you can see over time it increases. In 1950, it just takes off. It goes up really high. And so the question is, what does that mean? And normally, as I said, if I was working on a temperate lake, I'd be like, oh my goodness, the lake's becoming eutrophic. And I'd be looking for like a leaky septic system or something. What do I mean when I see eutrophic? Eutrophic typically means that we're getting increased algal production because of increased nutrients. Could be nitrogen, could be phosphorus. Why do we care about eutrophication? It can cause all kinds of problems: loss of biodiversity, loss of oxygen in the deep waters, degradation of water quality, changes in water taste, water odor, fish kills, the list goes on. And maybe some of you have heard about Lake Erie and recent algal blooms in Lake Erie. That's another thing that can happen with eutrophication, is algal blooms and toxins associated with algal blooms. So again, I was like, why is this showing up in these lakes? And over the years, I've noticed, and I think people working with me, we wondered perhaps that algal production's increasing. But again, we don't have any, well, we have some, I should say, but we don't have measurements going very far back in time, not more than 10 years anyway. So the question is, has algal production changed? If these lakes are becoming eutrophic, we should expect that algal production is increasing. So these were the questions that I started with. What is causing these diatoms to increase? Has algal production changed over time? And I think these are important questions to think about, because if you want to protect these lakes, you need to know what's driving changes. So as I said, a series of questions came to mind. Has algal production actually increased? What nutrients driving it? Is it nitrogen? Is it phosphorus? Is it both? What's driving the changes in nutrient inputs, if there are changes in nutrient inputs? And is there possibly a climate link to all of this? So we started by thinking about, well, what causes eutrophication and what would cause eutrophication in an alpine lake? So first thing is increased nutrients, nitrogen, phosphorus. What could cause that? Maybe atmospheric deposition, because there's nothing happening, or it doesn't appear to be much happening in the catchment. What would cause changes in atmospheric deposition? Increased fossil fuel burning could cause increased nitrogen deposition. Agriculture dust could lead to increased nitrogen or phosphorus. Phosphate mining could cause increased phosphorus. Then we thought, well, maybe grazing. There's these lakes, the catchments of these lakes have been grazed. So maybe if you had erosion occurring, you would cause some nutrients to move from the catchment into the lake. Maybe that's what's causing it. What about fish stocking? Could that cause a change in nutrients to the lake? And then what about warming? It's probably not going to directly change nutrients, but could it somehow cause eutrophication? So we started, I should also say, we also thought about, are alpine lakes particularly sensitive to these sorts of changes? And in particular, when you think about atmospheric deposition, they should be pretty sensitive because there's not really a lot of soil development. Vegetation tends to be relatively sparse in a lot of particularly alpine environments. And that means that if you have nutrients coming from the atmosphere landing on the catchment of a lake, there's nothing there to use them. So they're all going to end up in the lake. So that suggests they could be quite sensitive. We also thought there's lots of snow. So over the winter, you could have a lot of nutrients accumulate and then get this big pulse in the spring, which could lead to a spring bloom of algae. So those are things we thought about at the start of the project. And the first thing we looked at was fish stocking and whether fish could cause a change in nutrient loads in lakes. So previous research from the Canadian Rockies, and this is just one example, there's other studies out there, indicated that you could have 4 to 10 times more algal production after you stocked a lake. And there's a couple ways this could happen. One is that if you stock a lake, fish can sometimes move nutrients from the nearshore environment to the open water environment. And that could cause changes in the algae living there and could also promote algal growth. It could also be caused by a change in grazing. So if you add fish and the fish eat the zooplankton, then there's nothing to eat the algae and that you could see an algal bloom. This particular study, which was done by a fellow named Peter Levitt, they did some experiments with nutrients and they were pretty convinced that the change was mostly related to increased phosphorus. So we thought, let's kind of do the same study they did and see what we find in the Uinta Mountains. So we looked at 3 lakes here. This is on the north slope over here. And Birdsong Lake is a fishless lake. It's our control lake. It's never been stocked. Hidden Lake North and Justin Lake both have been stocked. You can see Birdsong here, Justin Lake here, and Hidden Lake here. They're very similar in terms of their size, in terms of their catchment area. The only real difference is that one has never been stocked and the other 2 have. So here's a little bit of history for you. Fish stocking in the Uinta Mountains, I would say officially started in the 1950s, but there's a good chance that started even earlier than that. Most of the lakes in the Uinta Mountains, and in fact in most of the western US, were fishless. And this is mostly because there's natural barriers. In fact, some people have estimated that like 95% of alpine lakes in the western US were fishless. So adding fish was a big change. The other thing that happened in the Uinta Mountains was grazing, which probably started around 1890s and peaked in the 1920s to 1930s. And then they started to reduce how widespread grazing was occurring. And so let's take a look at the results and what we found in these 3 lakes. This is Birdsong Lake at the top, Justin Lake and Hidden Lake. These are just pictures of the 3 lakes. What I just want you to look at, it's like a really busy diagram here, but this is time on the y-axis. So going from the top is today and going back to about 1600. And then what you see, we'll just look at the green, the blue, and the orange curve. We won't worry too much about what they are exactly are measuring, but they're all showing algal production. And what you can see in all 3 lakes is that about between 1940 and 1960, that they increase. So first thing to take from this, the timing rules out grazing, because grazing started earlier than that. It peaked in 1920 to 1930. By 1940 to 1960, we would expect to see, if it was algal production driven by nutrients related to grazing, we'd expect to see it going down, not up. So we don't see that. It does match with fish stocking. 1940, 1960 is pretty close to 1950, but our fishless lake shows the same changes as our stocked lakes. So that suggests it's something besides fish that's driving this increase in algal production. So first study suggests that grazing's out because the timing's wrong. All 3 lakes are showing changes in algal production. So that suggests it's not fish stocking that's really driving the main change. I don't want to say fish stocking isn't having any impact, because maybe it's just being masked. It's too small to be seen by whatever is driving the main change. So we can rule out grazing. We can rule out fish stocking. Now what? We'll look at atmospheric deposition of nitrogen. So why am I thinking about nitrogen? Why would I expect nitrogen to increase from the atmosphere? And humans have increased what we call reactive nitrogen. So that's nitrogen available for algae and organisms by about 2 times what it was before humans started burning fossil fuels and using synthetic fertilizers. So we've really changed the nitrogen cycle, and global deposition has also changed. So this is a good example of a global change problem. And the dark reds show where we have higher amounts of nitrogen deposition on a global scale. So we wanted to see if these lakes were perhaps changing in response to nitrogen. So these are 4 different lakes now on the south slope, and the study actually had 6, but I'm just going to show you 4 for tonight. And again, another busy diagram, but we're just going to look at a few things. So there's 4 rows. Each row is a different lake. This is showing you the mud from those 4 different lakes. This is time. Today's at the top, 1600 or so at the bottom. It's a little different for each one. And this is showing our Asterianella formosa diatom. And you can see it increases, but at different levels and slightly different times in all 4 lakes. And this is our algal production, which shows an increase in all 4 lakes. The increase in algal production is about 1940 to 1960. And oops, I went too far. Anyway, it matches pretty nicely with the Asterianella formosa. Now, the other thing I want to draw your attention to is the nitrogen isotopes. So these are stable isotopes of nitrogen. There's one, I'll just remind you, isotopes are, they have the same atomic mass, or sorry, same number of protons, but different atomic mass. And if we compare their ratio, it can tell us about nitrogen sources, for example. I'll maybe talk a little bit more about that in a minute, but this is just what you'll notice is when we see changes in Asterianella formosa, we also tend to see a decrease in the nitrogen isotopes. Not always, but generally. And if it was driven by fish stocking, we would expect actually the opposite. So this is more evidence that fish stocking isn't our problem. If it was related to synthetic nitrogen fertilizer or fossil fuel combustion, we would expect it to decrease. Now, there are other things that could cause nitrogen isotopes to decrease. I'm not going to talk about that tonight, and I'll kind of walk you through some other data to show you that that it could very well be, we think it is, related to synthetic nitrogen fertilizer and fossil fuel combustion coming in atmospherically. So the other question that we had was, is there anything in timing of potential sources of nitrogen that would support this idea? So we looked at population along the Wasatch Front, which is to, like where Salt Lake City is, and is to the west of the Uinta Mountains, and prevailing winds are probably coming west to from the northwest and would bring pollution from the Wasatch Front. So as population increased, so did fossil fuel burning. And so this potentially could increase nitrate deposition in the Uinta Mountains. We also looked at nitrogen fertilizer and its use along the Wasatch Front, and it generally increases by beginning 1940 to 1950. And we can see population really starts to take off around 1950 as well. So yes, there are sources, potential sources that could explain an increase in nitrogen. But there are also sources that could suggest an increase in phosphorus. So then we were back to this, well, it looks like it's a atmospheric deposition of nitrogen or potentially phosphorus. So we thought again, we went back to our diatoms, Asterianella formosa, and we collected samples and environmental measurements at 65 different lakes, and we put it all into a fancy statistical package and said, okay, tell us what environmental variable is controlling the distribution of Asterianella formosa. And we got out, I won't bother going into big details about this diagram, but the result was that nitrate and Asterianella formosa were highly correlated. So we were like, oh, well, maybe it is nitrate then. And then we saw a nice study written by a woman named Jasmine Saros, and she had done lab experiments that showed that Asterianella formosa was very sensitive to changes in nitrogen, and that probably was what would cause Asterianella formosa to increase nitrogen in lakes. So that got us thinking, where's that nitrogen coming from? We sort of alluded to the idea, maybe it's fossil fuel burning, maybe it's fertilizers. I wanted to find out where it was coming from. So I teamed up with Professor Fred Longstaff here at Western, who runs the Laboratory for Stable Isotope Science. And his student, Sam Russell, paired up with my student, Beth Hundy, and they came up with a way to look at where the nitrogen in the Uinta Mountain lakes was coming from by measuring nitrogen and oxygen isotopes in nitrates in the water. And this is actually not an easy thing to do, particularly in the Uinta Mountain lakes, where the nitrogen concentrations are still relatively low. They've increased, but they're still low. So basically, Beth collected water samples from inflows, from snow, from the precipitation, from the lakes themselves. And then with Sam, measured the stable isotopes. And remember, these values here are showing you the stable isotopes. They're just ratios. So N15, you're looking at N15 to N14. Oxygen 18, you're looking at O18 to O16. And a variety of chemical processes will cause those ratios to shift. So knowing the values of those can tell us a lot about how, where the nitrates coming from, or what chemical processes it's gone through before it's become part of the precipitation or the lake water. And then we use some fancy statistics, stable isotope analysis in R, and Bayesian isotopes to figure out exactly where the nitrates came from. So just a quick, I'm going to be going over time here, I think. Quick little bit of background. Stable, if we look at nitrate, that's NO3. So it has nitrogen in it, it has oxygen in it. And oxygen in the environment, we have 3 stable isotopes, O16, O18, and O17. Most of it's O16 and O18. And the stable isotopes of nitrogen are N14, most of it, and a little bit of N15. And as I said, those ratios, if we compare O16 to O18 or N14 to N15, they tell us about source. So what we did, I should say what Beth did, which is different, was also to look at O17. And O17 relative to O18 is very interesting. Basically, people don't usually measure O17 because when like say water or nitrate is formed on the Earth's surface, it is always in the same ratio, O17 to O18. And so the values will always lie on this line. However, if it's formed or is part of a chemical that's formed in the atmosphere, like ozone in particular, then O17 will be enriched. And so this enrichment we measure as the difference between our value 017 to 018 compared to what it would be if we were right, if it was formed on the Earth. And we call this cap delta 17. So that's the difference between if it was formed on Earth versus if it was formed up in the atmosphere. So whenever we have things that aren't on this line, when we measure our 018 to 017, it tells us that the nitrate is formed in the atmosphere. So here's our line. So if our points, these are all Uinta samples, and I'll talk about them in a minute, but if they were to lie on this line, it would tell us the nitrate have formed in the terrestrial system down here. If it falls above the line, it means the nitrate comes from the atmosphere. And the green triangles are showing you snow, and the orange squares are showing inflows. The blue diamonds are lake samples, and the purple is snow melt. So what you see is the snow is almost all atmospheric, or not almost all, but a large component is from the atmosphere. Whereas we come down to inflows and lakes, we're still off that line. So it's still composed atmospherically, but less so than the snow. The next thing we can do is plot them with our cap delta 017. So that's the difference between, let me just go back for a minute. That's this distance. And this is telling us about our nitrogen isotopes here. And if it's coming from agriculture sources, it's going to be plotting over here where this blue bar is. If it's coming from, say, septic systems or manure, it's going to be plotting over here. So let's take a look at our Uinta Mountain samples. These are the snow and snowmelt samples. So again, plotting high up, this is where this purple box is where we'd expect to find atmospherically oxidized nitrate. So nitrate formed in the atmosphere, and they're landing close to that. This is where our inflow and lake samples are plotting. So less of it is formed atmospherically, but certainly a component is formed atmospherically, and it's mainly plotting in the fertilizer, but also a little bit into the soil, which makes sense if you're having some come atmospherically from distant agriculture and some coming from the soils within the catchment. So take-home message is that when we ran this through the Bayesian statistics, about 23% was atmospherically oxidized nitrogen, 62, and that's can be formed either from lightning or from fossil fuel combustion. 62% was coming from fertilizers, 13% was coming from catchment soils, and pretty much nothing was coming from manure. So is this just a Uinta mountain lake phenomenon? No, we compared it to other oxygen and nitrogen values. People don't normally measure 017, but based on our 018 and nitrogen values, probably most mountain ranges in the Western US, these different colors represent different mountain ranges in the Southwest US, are receiving some component of nitrates from atmospheric deposition, either from fossil fuel combustion or from agricultural sources. So findings, we estimate that 10% of total nitrate inputs are from fossil fuel combustion. 62% of nitrates are from fertilizers used in agriculture. So I'm really, where am I? I'll just take 2 minutes and tell you a tiny, I'm okay? Yell at me to stop. So atmospheric deposition of nitrogen is a player. Last one I'll look at just very quickly is warming temperatures. And we know that warming is occurring in the US, and particularly in the Western US. These maps are showing changes in temperature between 1986 and 2016 compared to 1901 to 1960. So the red colors are saying it's warmer now than it was in the past. And here's our Uinta Mountains, bright red. Temperature change in that time period is more than 1.5 degrees. These are showing winter, cold temperatures, and warmest temperatures. And again, you can see the Uinta Mountains are seeing big increases in the warmest days, or the temperature of the warmest day of the year. So warming is happening across the US, particularly in the West, and the Uinta Mountains are definitely being impacted. So Asteronella formosa, I told you it likes nitrogen. It also is adapted to thermally stable waters, which we expect to see increase as temperatures warm. And the reason they're adapted, they're really good at floating. So thermally stratified waters tend to mix less. So diatoms that are good floaters can stay up in the photic zone better. And so that's a good competitive advantage to have. So we went back to our 3 lakes, Birdsong Lake, Hidden Lake, and Jessen Lake over on the north side. And we also looked at temperature records and nitrogen deposition and fertilizer records. So we only have nitrogen deposition records from the NADP, National Atmospheric Deposition, going back to about 1980. But we looked at fertilizer use in the United States and actually in Utah too. And what you can see here is it really takes off when we start using synthetic fertilizers in the late 1950s. And you can see it matches up really nicely with nitrogen deposition. And this is showing temperature from 1900. I should have said this is 1900 to today, 1900 to today. And you can see fairly stable temperatures. There's some ups and downs. And then we see this increased temperature pretty consistently from 1980 onward. This blue line is statistically determined as when there's a big change in temperature direction. And again, showing this increase. And I showed this with this little blue arrow. And we just said this is the break point. We didn't have enough data back here to determine statistically where the big change in nitrogen occurred, but it's about here in the 1950s or so. So 2 points, temperature change, late 1970s, 1980, nitrogen change, 1950. And then we looked at our algal production from those 3 lakes. So you've seen these curves before, going from 1980 to 2000, 1980 to 2000, 1900 to 2020. Sorry about that. And looking at chlorophyll A over time, this is our control lake. We can see it increasing, increasing, increasing. And the orange and blue arrows are showing you when nitrate goes up, when temperatures go up. And we did the same statistics on the chlorophyll A, and we can see that chlorophyll A, the break points, time that the chlorophyll A is making big changes, occur just after these arrows in all 3 lakes. So that suggests that both nitrate deposition and warming temperatures are driving algal production. We don't totally understand the mechanisms that are driving algal production related to temperature. So we're working on that still. Answers to the questions we started with. When did algal production increase? 1940 to 1960. Which nutrient is the culprit? Nitrogen or phosphorus? Nitrogen for sure, but it could be both. What's driving the change in nutrient inputs? Specifically nitrogen, atmospheric deposition from fertilizer. There's new research going on to look more closely at phosphorus, not by me, but by another woman, Janice Brainey at Utah State. And is there a climate link? Probably. So we are working, as I said, to try and better understand the connections between warming temperatures and algal production changes. Using, rather than just paleolimnology, now we're adding a limnological approach and monitoring both lake water temperature and water chemistry. So, conclusion, fish stocking has small effects in the Uinta Mountains. Remote alpine lake ecosystems are changing as a result of long distance transport of nitrogen, originating mostly 70% from fertilizers and climate warming to some extent. These are global changes. So we're learning about them by studying mountain lakes in the Uinta Mountains. Tough challenges. How do you protect lakes from global change? It's not as easy as dealing with fish stocking. So what do? I don't have any good answers for you, but it's, I think, important to think about this and important to think about what we can do individually to try and protect these lakes. So special thanks to funding agencies. Also to John Small and Brian Cumming at Pearl, Queen's University, for providing chlorophyll A analysis and also helping with some of the dating. And most important, as you might imagine, I didn't do this by myself. Thanks to all the people who've helped me over the years to collect this data and do lots of lab analyses. I've had an amazing group of students over the years. So thank you very much. And with that, I'll take any questions. I'm sorry I went a bit over.
That's wonderful. Thanks again, Katrina. And yeah, you mentioned what action people can take on a personal level. Is there anything that folks can do personally to make a difference?
Yeah, so that's a really good question. And what I always encourage people to do is write letters. It does make a difference. Sometimes it's only small differences. But, and I can speak less for the US situation, but in Canada, politicians, we had someone come and give a talk at my class, and they said that the Minister of the Environment received only 40 letters in a year. And so they pay attention to those letters because that's the only input they get about what should be done. Right. So writing thoughtful letters to government agencies, asking for change, telling them what you want to see changed and why is really important. I also think individual actions make a difference. So anything you can do and set an example for change, whether it's reducing the amount of plastic you use or riding your bike to work, whatever you can do sets an example. And also I think helps you take further action, like writing letters. It's a positive feedback, I think. So those may not seem like much, but I do think anything individually you can do to help protect the environment makes a difference.
Absolutely, thank you. Ed submitted a few questions, one of which is, what will these lakes look like in 15 years?
Oh, I don't like that question. So good question. So I don't like to be negative, but it's hard not to be. Everything's pointing to increased algal production. And so is that what does that mean? It means that you're changing the ecosystem from what it is. I'm not going to say it's a good thing or a bad thing. These are traditionally very, very low productive systems, and you're going to make them productive. So if you're a fishing person, maybe you'll think that's a good thing, right? Because now maybe those fish will survive a little longer than they do right now. But you're changing a system that's been relatively unchanged for hundreds to even thousands of years to something different. And to me, that always sounds warning bells. I don't think these are going to become like Lake Erie with toxic algal blooms anytime soon. They're still very low nutrients, but they are very sensitive to even small nutrient changes. So they will be different and they'll continue, I'm sure, to be changing, and you will see more algal production, I'm sure.
Mm-hmm. As a follow-up to that, Ed asks, and thank you, Ed, for these great questions. Are there changes in agriculture on the horizon that will have impacts?
Yeah, so that's a really good question too. And I would say yes. I mean, there is things already happening in terms of being applying fertilizers more, sorry, I'm blanking out on the word I want, but more carefully so there's less waste. And it's a bit, you know, the agriculture thing's tricky too, because we need to be very productive with our agriculture. We need to be able to produce as much as possible. And that's one of the reasons we use fertilizers, but we also tend to overuse fertilizers. So trying to reduce what we use will make a difference. And I have no doubt, like, we're really good at technological changes. So I have no doubt that we'll continue to improve that, especially if people are aware of problems like this. So again, I like to be optimistic about things.
A couple of linked questions. Donna asks, how do fertilizer components get into the atmosphere? And Bob asks, are the sources of nitrogen from fertilizers from areas in Utah, or could it be from California or even further away?
So I'm going to start with the second question first.
Sure.
And they're both very good questions. We don't know. I talked about the Wasatch Front as one possible place. It could be California. It could be even farther afield. We looked at how far nitrates can be carried atmospherically, and it probably isn't too much farther than California, but it's possible it could be a bit farther than that. How does the nitrates get into the atmosphere was the other question. So there's actually several different ways. When it's applied as a fertilizer, it can be just carried as dust. It can also be volatilized and then altered in the atmosphere and precipitated out. So there's a couple of different ways that it can be carried.
Interesting, thank you. Ed also asks, is there any evidence that Oregon lakes are similar to those that you've studied?
Yeah, that's a good question too. I don't know the answer to that actually, but I can tell you I have worked in the Sierra Nevada, and that system appears to be different. And I haven't looked specifically at nitrogen. I was working in the Sierra Nevada to look at changes in drought over time, but we didn't see the same kind of relationship that we saw in the Uinta Mountains with aster and formosa and with nitrate. And it's kind of interesting because I did the Sierra Nevada work first and then wanted to reconstruct drought in the Uinta Mountains, and it didn't work at all. The reason it didn't work is because the high elevation lakes aren't responding to changes in moisture at all, or even they're just totally messed up by the nitrate. So you don't get this clean climate signal out like we did in the Sierra Nevada. So I don't think the Sierra Nevada is affected the same way as the Uinta Mountains, but the Colorado front is, the Teton Mountains are, several other ranges are, but Oregon, I don't know. And we didn't have that in our sample set. So I don't know. And I don't know off the top of my head if anyone's worked on that. And I don't know what the nitrate deposition levels are like there either off the top of my head. It's a good question though.
It was, thank you. Another great question from Bob this time. Could there have been any impact from nuclear tests in the late 40s and 50s? That's a great question, Bob.
It is a good question. So you're not talking about nitrate, though. You're just talking generally if they've affected the lakes at all. Is that right?
That's my guess too. I'll wait and see if
Yeah.
does.
Okay. Yeah, not that I don't know. And we haven't looked at anything that would answer your question, actually.
Katrina, what's been the most challenging aspect of this work?
So I don't know that it comes out in the slides because it always looks fun, right? And that's because when I take photos, it's usually nice out. But the weather can be quite challenging, and it is physically demanding, the work, for sure. And unfortunately, I'm not getting younger, so it gets harder every year. And my students are always 20, so I would say that has been the most challenging. It's physically getting the samples is tough. And I have to say, there's been folks from the Forest Service who, I think some of them might be in the audience, have been a huge help to get samples. So yeah, but I would say that's been the toughest part. It's beautiful though, and I was saying to Louise before we started that because of COVID, I haven't been there for a year and a half now, and I miss it a lot. So
Wonderful. Well, I think that's it for questions from the audience, Katrina. I'll let people, this is your last chance, folks. If you have any final questions, we've got some thank yous coming in for you, Katrina. Bob says, thank you, this is very informative. And I echo that sentiment right there. Give you a minute in case you have any last burning questions to ask Katrina. Katrina, what's next for you? What are you studying next? What's your focus moving forward?
So, well, I'm working on projects up in the Canadian Arctic and also locally where we've got projects and both of those studies, both in the Arctic and here in Southern Ontario, are related to climate change. That's been my main research focus. And as I said, that was what I was supposed to be working on in the Uinta Mountains, and then it got sidetracked to thinking about algal production and nutrients. So yeah.
Wonderful. Well, thank you again for joining us this evening, especially so late your time.
Oh, no problem.
And thank you all for attending this Natural History Pub and for all your great questions too. Just one final thing before you go. As part of being a finalist for the 2021 National Medal, the Institute of Museum and Library Services will feature the good work of the High Desert Museum this Thursday, April 8th, both on its Facebook and its Twitter feeds. We will spend the week shining, and we invite you to shine with us. Please share your stories about what the High Desert Museum means to you on Facebook and Twitter. You can tag US IMLS and include IMLS medals to help the museum out and celebrate our work. And again, our members make this possible, and our sponsors too. So we're grateful for all of you. And with that, I'll say good evening and thanks.